Okay, how are we doing in year 11? Today's lesson, okay, we're going to start off with a bit of knowledge recall. And you remember from our last lesson, we were looking at the process of protein synthesis, quite a tricky topic. And we did a lot of works. Some of you were using diagrams, some of you were using more of a storyboard to describe the process of protein synthesis. So we'll start off with some knowledge recall today. Just looking at these images. Now, ordinarily, I'm going to be working in pairs and using these images to describe to each other the process of protein synthesis that we looked at in our last lesson. We're also going to be doing this at home for this particular lesson, so I'm just going to spend some time looking at each of these images and trying to record as much as you can about each of the images. They are in order of the process. So the first one, we can start off together. So first of all, first step in protein synthesis is that the DNA strand should unwind and a template is made. Okay, so take that as your starting point. Try to recall from the images as much as you can. Okay, pause the video, have a go. So once you've done that, you can see here this is what we used as part of our sort of storyboarding to describe the process of protein synthesis. So you can have a look through this and just think about what areas did you not forget, what areas did you absolutely nail. So first of all, a template is produced that reflects the sequence of the bases in the DNA. So depending on the protein that we're going to make will depend on the template that we're going to make. And it might be that just part of that DNA, part of that double helix will unwind to reveal a section of DNA which we call gene. I'm going to go back over these terms in a bit more knowledge recall in a minute. So we make the template is small enough to leave the nucleus, because we can't make new proteins within the nucleus itself. That's another reason why we make that template. The template moves out of the nucleus through these nuclear pores and binds to a ribosome, which will be in the cytoplasm. And it's here in the cytoplasm that we can make these new proteins according to the genetic code. These carrier molecules in the cytoplasm, each attached to a specific amino acid. Okay, remember 3 bases. Code for one codon, okay, which is one amino acid. Attach themselves to the template in order to give the DNA given by DNA until the template is completely ready. It might be that sequence of DNA is a couple of hundred bases long, it might be 1,000 bases long, whatever it is, it will stop until that DNA code is read and the protein is made. As each of those codons gets read, each of those codons brings the amino acids to the ribosome, the amino acids will join together to form a specific protein, and initially we'll call that a polypeptide chain. The protein detaches from the carrier molecules, and each will detach from the template and return to the cytoplasm to pick up more amino acids, and that will keep on happening. Once the protein chain is complete, the molecule folds up to form its unique shape and will enable it to carry out its functions. And it might be that a new antibody is made, or a new enzyme is made, or whatever it will be, whatever type of protein it will be, whatever it folds up to make is that specific protein. So think about how much do we remember from that process. Pause the video. How much do we remember? Okay, on to the next slide. So we've used lots of key terms in introducing DNA and DNA structure and protein synthesis. I just want to use this diagram to kind of clarify the difference between the terms we've used. We know that the DNA, we've referred to genetic material and DNA being found in the nucleus. We have referred to chromosomes, we've referred to DNA, we've said genes. What is the difference between all of these terms? And we see here nicely that clearly we know that the nucleus is where all of this genetic material is found. The nucleus is inside our cells. Inside, I said in the example, if we took a skin cell, for example, you'd find all of the DNA needed in that nucleus to create an exact copy of you. And all of the DNA is located on these structures called chromosomes. Okay? Tightly packed DNA into these structures called chromosomes, which you can see labelled here. Now, I'm not going to talk too much about chromosomes just yet, because when we look at reproduction and inheritance, we'll learn more about what chromosomes are. But for now, we just need to know that that's where the DNA is located, on these chromosomes inside the nucleus. Okay? We have a total of 23 pairs of chromosomes, 46 in total, half from mum, half from dad, that make up us, who we are. Now, if we were to unwind all of that DNA from the chromosomes, a section of that DNA would be called a gene. And it's the DNA then that unwinds to reveal the base pairs that's then made a template to then code for amino acids to then make proteins. So DNA, this is all DNA here, all of this lovely double helix, that is all DNA. The section of the DNA that is read is called a gene. Okay? A gene codes for specific proteins that make us who we are. Now today we're going to think a bit more in detail about gene expression, so the outward expression of those genes, which we refer to as the phenotype. We'll use that to come back to that key term when we look at inheritance. So the expression of these genes and mutations, what happens when we have a mutation in the genetic code, which can be caused by various things, various environmental factors, various things that we encounter can cause mutations, but they do occur naturally as well, causing mutations. Now, are all mutations harmful? I want you to pause the video and have a little think about that question. So please make sure you've got your title in your books. Gene expression and mutation, that's the focus for today. You need to be able to describe what mutation is, explain why the correct folding of a protein is important to its function, and outline the reasons why most mutations are actually harmless, because they happen a lot of the time. This is the link to the specification. Those that are interested in the specification, you can have a read through that and pause the video. And here we're going to get started by just thinking about the basics of our gene expression ideas. So here's a straightforward kind of recap. We're just trying to think about the words that are missing here, and then you can write that into your books. So when a gene codes for a protein that is synthesized in the cell, the gene is said to be expressed. Okay, so when it actually codes for the protein synthesized in the cell, we said that the gene has been expressed. It's done its job. The gene has been read, it's made the protein that's been expressed. Most of the DNA in your body does not actually code for proteins. A lot of the DNA in your body is what we call non-coding DNA. So DNA that doesn't code for proteins is called non-coding DNA. And a lot of that DNA is just there to kind of keep you alive. Keep you alive, we often refer to it as a kind of blueprint of life. Although scientists don't know much about non-coding DNA, they do know that it is involved in switching genes on or off. And that's another key role of non-coding DNA, is to turn genes on and off, okay, to allow them to be expressed or to allow them to not be expressed. So let's make sure we have this written down as an introductory paragraph in our books under the title. Pause the video if you need to have a break. Okay, we've got a short video here. You might be able to just copy this into your browser if you want to actually have a look at the video. We'll play this as well here.
When you think of the word mutation, it probably isn't a surprise that some strange or not so pleasant ideas come to mind. It's easy to get caught in the belief that all mutations are bad. However, be not afraid. What a mutation really is simply a change in your DNA sequence. You may be wondering, how did these changes even come about? Mutations can either be inherited, meaning passed down in the sex cells of the parents, or random, meaning that they spontaneously appear in an individual. Mutations come in many different shapes and sizes, as shown by researchers at the U.S. National Library of Medicine. For example, MISSENSE mutations are a change in a single DNA base pair, causing a substitution of an amino acid. Frameshift mutations happen when DNA base pairs are added or removed, resulting in a change in the reading of DNA during the copying process. Deletion mutations result in the removal of a piece of DNA, which will in turn affect the function of the protein that is later formed. These are just a few examples, and there are many, many more different mutation types. Now, what can these mutations do to you? Or in other words, what effects can they have on phenotype? Of course, the most well-known category of all are the harmful mutations. They usually result in a disease or disorder, which can sometimes even lead to death. According to an article published by Erickson in 2010, an example of a harmful gene mutation is one that results in paroxysmal nocturnal hemoglobinuria, which causes breakdown of red blood cells, thrombotic complications, and death in 50% of cases. While that may be a bit of a downer, mutations can also be neutral or even beneficial. Neutral mutations occur in an area of DNA that do not get translated into genes, which means that they are not capable of causing any changes in an individual. Beneficial mutations are those that typically are passed into offspring because they offer an advantage that can increase their survival. A good example of this is when a mutation arises in a gene that can offer resistance to a disease. For example, as explained by Carrington et al. In 1997, there are gene mutations that can increase an individual's resistance to infection of the HIV virus, reducing their chances of contracting AIDS. So why is it that knowledge of harmful mutations is so popular, while no one ever really mentions neutral or beneficial mutations? The misconception stems from the fact that people who have a disease typically seek out genome analyses, while those that have neutral mutations can go their entire life without knowing, and so never get tested. Keep in mind that even though a bad phenotype is not expressed, it does not mean that you don't have any mutations in your DNA. Harmful mutations have unfortunately given all mutations a bad rap, although this really isn't the case. Remember that your mutations are not necessarily hurting you, and having mutations does not make you a mutant.
Okay, so really good video there. Hopefully the audio should have come through on that video as well, but if not, you can see the link here and do have a look at that video. This is just looking back to the original question at the start of the PowerPoint, which is, are all mutations bad? And the answer is no. Some of them, there are harmful mutations, and depending on how that affects your genetic code, will depend on whether that is a harmful mutation or not. But also, there are some useful mutations, and it's mentioned in the video there about mutations that can lead to resistance and disease, and clearly that's a good thing. So there are different types of mutations, and not all of those mutations are bad. And of course, some can be completely neutral. It might be that the DNA sequence, there is a change in the base of that sequence, but it may still, as a whole codon, code the same or a very similar amino acid. So we can have neutral or no effects in mutations, because mutations are quite frequent.
When you think of the word mutation,
so this is an example that we're going to kind of illustrate and draw into our books and think about the possibilities. Okay, so picture one, what we've got here on our diagrams here is our chromosomes, just represented quite crudely, the pair of chromosomes and the pair of genes there located as dark pink. And the genes there are coding for a particular enzyme. Alright, you can see that a mutation has occurred and that gene has changed. Alright, so picture one is a normal gene which codes for an enzyme. And this enzyme has an active site, that's where substrate binds to. That is a complementary shape to the substrate. You can see that here with the enzyme here fit as the active site and the substrate fits perfectly there. Picture two is the same gene, but it has a mutation. And I wanted to pause a bit now and think about what are the three possibilities, or three possible types of mutations that could arise from this change. So we've got to imagine here, use our imagination here, that a gene has undergone a mutation, whether that's an insertion of a new base, or whether there's a deletion of a base, whether the base is changed to another base to code for a different amino acid, whatever it may be. The result is that there could be some changes to this particular enzyme. And we know that enzymes are very important to control our reactions in our body, our biological reactions. They are, of course, biological catalysts and are a protein. So if the sequence that makes this enzyme changes, what could the three possible outcomes be? Okay, so pause a bit, have a look. So here we've got our three possibilities. First of all, the enzyme could be completely unchanged. Just like the video, there would be a neutral mutation that doesn't affect, profoundly enough, the amino acid sequence, the protein that makes this particular enzyme. So the enzyme would be unchanged, okay, so almost unchanged. You see it's slightly different shape, looks slightly different, but still is able to work effectively. So almost a neutral mutation. The second possible outcome might be that the active site, the shape has changed so much that the substrate can no longer fit, and therefore the enzyme can no longer do its job. That could be a really really profound problem if we no longer have this enzyme that does its job and could affect the biological reactions in the body. So we've got number 1 unchanged, number 2 the active site is changed, the substrate can no longer fit, and number 3 it might be the active site changes shape so that a different substrate can now fit, and so it catalyses a different reaction. Okay, so we've got a nice little example here of different types of mutations and the different outcomes that can happen. The first one here, number 1, is referring to what we call the neutral mutations. Number 2, there is a change that is harmful, potentially. And number 3, there is a change that could possibly be beneficial. So I'd like you to listen back to this part of the video, draw this into your books, and add the 3 types of mutations here. Neutral, harmful, and beneficial. 1, 2, and 3. Pause the video and draw the diagrams and annotate as follows. Okay, what we're going to do now is have a go at some questions. Now, unfortunately, we don't have the textbooks with you, which is a shame, but what I'd like you to do is just have a think about these first 2 questions. Okay? I really want to spend some time thinking about the question in green, suggests ways in which the expression of a gene may be controlled, and the question in pink, explain how mutation can change the appearance of a protein. I want you to think about these 2 questions in particular, and you might want to pause the video and just spend a bit of time doing some independent research, okay, so we don't have the textbooks with us, doing some independent research of those 2 particular questions. Alright, you might want to jot some notes down, but you just have a think about these 2 questions, the one in the green and the one in the pink. Those of you that are really, really looking to stretch yourself, you can also have a go at the 6-mark question here as well. So pause the video, do some research on the green and the pink question, and then we'll start having a look at some model answers and write those into our books as well. So pause the video, do some research and have a think of those first 2 questions. Okay, so we're going to go through the answers here, and if you weren't too sure, see how close you got. You might want to use this now to write a model answer into your books, please. So suggest ways in which the expression of the gene may be controlled. Well, non-coding regions of DNA, that's what we're referring to here, involved in switching genes on so they are expressed, and switching them off so they're not expressed, or switching only part of the gene on so different proteins are expressed. And this idea really is based around this idea of the job of the non-coding DNA. Explain how a mutation can change the appearance of a protein. Well, the order of bases in DNA controls proteins made on ribosomes in a cell. If the mutation alters the DNA bases, this may change amino acids joined together to form the protein. But it may not. It needs to be recognised that. This affects the way in which the protein folds and functions. If an enzyme, we used that example before, if an enzyme cannot work, certain chemicals will not be made, and this can affect the organism's appearance. If mutation affects the non-coding DNA, the gene expression is affected, which would also affect the appearance. So the key question here, explain how a mutation can change the appearance of a protein. A mutation is essentially changing the DNA sequence, which changes the amino acids, which therefore changes the protein. Okay? And that's the fundamental idea that we need to get our head around. So do use this now to write yourself a model answer using this mark scheme here for this question. Okay, and for those of you that were going for this real stretch question, you can have a read through this as well. Discuss how a mutation can have a good effect and bad effect on an organism. Use examples in your explanation. And you might want to go back through the video and remember the different types of mutations, ones that are neutral, ones that are positive, and ones that are negative. And you've got some really nice examples here. And we picked up on that idea of being resistant to viruses. That can, if we have a mutation that causes a resistance to something, a resistance to a disease, and that's a really good example of a positive mutation. Not all mutations are bad, relating back to our initial question. Lastly, a couple of good example questions to have a look at. And here's the mark scheme piece itself. Okay, thanks very much, Year 11. I'm going to post this video on the Teams channel, and you can work your way through this narrated PowerPoint, and then I'll be available on the Teams call as well towards the end of our lesson. Okay, thanks very much.